So it's a great pleasure to introduce Joe DeRisi, who is no stranger to Stanford. He actually got his Ph.D. here with Pat Brown, and but he's gone on to greater glory at UCSF and been a real pioneer in identifying pathogens through recombinant DNA and high-throughput sequencing technology, all kinds of cool stuff, which he will probably tell you about.
Thanks a lot. That was great. And thank you to the organizers, Lucy Shapiro and others, for having me here. This is an amazing group of speakers, and I'm really humbled to be a part of it and to tell you a little bit about what we're doing today. So thank you. This has been a great day so far. And I will be talking about this post-COVID autoimmune syndrome in children. But first, I want to give a little shout out to some of the things that we've been doing at the BioHub as I reflected on the earlier talks today. And one of the things that Dr. Birx brought up and others was the need for detection and surveillance. And that is something that we're really invested in through the Chan Zuckerberg BioHub in San Francisco for the last 8 years, headed up by Christina Tato, who's here in the audience over here, is being able to empower countries, institutions, hospitals, clinics, and labs around the world, and especially where the infectious disease burden is the heaviest, to use the latest in metagenomic technologies, which are not biased towards one pathogen, one parasite, one fungus, one bacteria, or another, but really can capture everything to get a better hold on what is happening locally and be able to transmit that information to a central place so that we can get an idea of what is happening globally. Why don't we have a pathogen weather map for the world? And so Christine and her team has been working on disseminating those technologies through direct training, hands-on mentorship in countries all over the world in a model in which trained labs can then train others, a train-the-trainer model. And then through CZI and CZ BioHub, providing compute, storage, and resources to eliminate the need to have a whole data science team in your university. And this with an eye towards a One Health network. As we heard before, One Health is super important. It can't just be humans. It's also livestock. It's also vectors. It's wildlife. And so many other things that go into this. We're all linked together. And so this is something we have a passion about. My interest in this began with SARS-CoV-1 when I started working on that in 2003 and helped in that response. And we were reflecting on where people were in January 2020. I was in Phnom Penh, Cambodia, setting up one of these sequencing nodes to try to detect emerging pathogens. And only a couple weeks later, it was the first Chinese tourist with SARS-CoV-2 in Cambodia, and it was picked up there. And so these networks need to be built and strengthened around the world. I am disappointed to see, as many probably are, that post-COVID, we aren't doing what we know we can do economically, cheaply, and effectively. And yet those systems aren't in place, and we still have fax machines in the lab. So like, what the hell is that about? Okay, so we've been doing this metagenomic technology not only for surveillance and all that stuff, but clinically as well. And we've been deploying that at UCSF for some time. There's also a spin-out called DevBio and many others in being able to bring this technology to bear on patient care. But there's a little bit of a dirty secret here, especially in the neurological infectious disease area, that is when we do this, there's a substantial fraction of patients who look like they have infection, but they don't. We can't find anything. And here's two examples here. This is two people. Both have been suffering from progressive ataxia. You can probably see trying to do the finger to the nose test or touch the finger and back, and neither one of them can do it particularly well. Both have a progressive ataxia accompanied with brain atrophy. And for all the world, which one has the virus and which one has an autoimmune disease? Because that's the other answer. If they don't have a virus or a bacteria or a parasite in their brain, and they have something like this, a meningitis or encephalitis, the other answer is it might be autoimmune. Now, I'm not going to talk about these two cases specifically, but I always hate not giving the answer. So the autoimmune disease is the one on the left, and the infection is the one on the right. And it's probably a virus you haven't even heard of, Cache Valley virus. But there are lots of viruses people have never heard about, but can kill you. So that's so the autoimmune part, the whole point of bringing this up is the autoimmune part is important, and we weren't really addressing that. Metagenomic sequencing or surveillance doesn't tell you if you have an autoimmune disease or not. And many of these autoimmune diseases maybe have linked or links to the underlying infection. Of course, we'll be talking about that with COVID shortly. Now, the poster child for autoimmune diseases in the brain is, I would argue, is anti-NMDA receptor encephalitis. So these are antibodies against the NMDAR receptor and can literally drive you crazy. There's a popular book written on this called Brain on Fire. It's a devastating disease, but now we know where the antibodies are and what receptor they go to can be tested for. But the reality is that's just one. That's one needle in a haystack. There's probably a lot more autoimmune disease linked to infection or cancers, in the case of NMDA receptor encephalitis or a teratoma. And we don't have good methods, or we need better methods to be able to screen them quickly and cheaply, to profile those antibodies, as well as T cells. And so how do we discover new autoimmune diseases? And by the way, these tests and doing these things in autoimmunity really also matter for pandemics. So one of the aspects of the pandemic that I won't have time to talk about here, but I'll just mention briefly, our colleagues, Jean-Laurent Casanova and ourselves, worked very hard on was the fact that anybody who had a pre-existing autoantibody to type 1 interferon was going to do very badly with SARS-CoV-2, and they did. They did very, very badly. They had terrible outcomes. And to this day, it frustrates me that we have no CLIA-approved test for an antibody to type 1 interferon, even though people are walking around with them, and we could predict who was going to die and who wasn't if they had that antibody, because your shields are missing. And so we need to do better on that too, because we know who's got these pre-existing conditions. At least we know who has how to test for them, but we don't have a clinically approved way to do it. And there certainly isn't a cheap Binax test for doing this either. So how do we go about discovering new autoimmune diseases when we don't know what they are? That was the easy case when we knew what they are, but what if we don't even know what they are? Well, there's lots of ways in the lab you can do that. You can do candidate-based approach, mass spec, protein arrays, lots and lots of ways. In my lab, what we thought about trying to attack the other side of the equation from the diseases we could detect with metagenomics was let's do something automation friendly. We need it to be inexpensive. We need it to be unbiased, high signal to noise, and highly scalable. And so some of there's only one technique that really fit all of those, but we supplemented it with others. And again, the population base here are those people we've already metagenomically sequenced with neurological disease that look like they have infection, but they don't. And we're going to take that cohort and go after them for autoantibodies. And so if it's neurology, we're going to do brain slice staining. That's not slow. It's not high throughput. It's not scalable. It's old school, but it does work. Immunoprecipitation mass spec, gold standard, really good, not very scalable, kind of expensive and slow, but important, and we still use it. And then there's programmable phage. And programmable phage has turned out to be a boon for us. So what is that? So this is a technique that is a variant of regular phage display. Phage displays got the Nobel Prize some number of years ago. And the programmable version of this was developed by Steve Elledge and Ben Larman back in 2011. And it was a technique that sort of sat on the shelf for a while. We adopted it and customized the heck out of it. Basically take all the protein isoforms of an organism, in this case the human, break it up into about 49 amino acid segments, clone those en masse into a phage system, and now you have a representation of the entire human proteome on phage T7. Not random peptides, but the human proteome overlapping. And so what do you do with that phage library? Well, imagine we have our unknown diseased patient sample, let's say it's CSF. We can combine their CSF, which has antibodies in it, with the phage library and capture those on magnetic beads featuring protein AG. Those bind the antibodies. So any phage that has a bit of the human proteome that binds an autoantibody will stick there on the beads, and one can then wash off the rest and then sequence the phage or repeat the process. And so it goes from library to incubation with serum. You can amplify those phage in E. coli, isolate the DNA, and then sequence it. So the beauty is we take a proteomic assay, immunoprecipitation with antibody, and convert it to a genomic assay using the cheap counting statistics of next-gen sequencing. That's the beauty of this. So it's dirt cheap, requires tiny amounts of material, semi-automated, we put on robots, very inexpensive, relatively speaking, and it's got a solid track record of success. So now I'm going to tell you a little bit features about the data, because this is a huge library, and for any given individual, you get a very complex profile out. And this will help you put into context the results when we talk about SARS. So first of all, the first thing you need to know about any individual when we take any antibodies from a person and do this procedure with is that myself and hopefully all of you here, you're all snowflakes. So if I take the entire profile of each person's immunoprecipitated peptides, that the autoreactome, if you will, and I correlate it with everybody else, you don't correlate with anybody. Everybody is themselves. They're a snowflake. That's kind of interesting, actually. And it follows from first principles. You all have different VDJ recombinations that make your antibodies. You all should be pretty unique. And it reflects your entire history of everything you've seen, which will be different. And even identical twins look different on this assay. So that's pretty interesting. One little side note, basically we can uniquely identify any individual with no genetic data, no DNA or RNA. Privacy laws have not caught up with that. Okay, so now the next question you may have is, if everybody's a snowflake, what happens over time? Is it just constantly a snowstorm, and every time you do this, you get a different answer? No. It turns out that individuals are incredibly stable, even over 92 months of time. That profile is like a rock. Even though they are encountering infections and other things in their lives, their overall autoreactome, the antibodies that react with their own peptides, with their own proteins, stays extremely stable. That you're a snowflake and it doesn't change. Just to add a little more reinforcement on that, what happens if you treated a patient with rituximab? Rituximab is a drug that kills a range of B cells. It's used as an immunosuppressant drug. Would the profile change? Sadly, no. It barely touches the profile. Rituximab itself is not powerful enough to eliminate the signal. These are myasthenia gravis patients treated with rituximab over time. Now, there is a medication that we know wipes out all B cells. That's anti-BCMA, B cell maturation antigen. So CAR T cells armed with anti-BCMA will obliterate all B cells in the body, including long-lived plasmablasts. And sure enough, when that's done, everybody reverts to being a new person. So 6 months after they've been treated, their autoreactome now looks different than what they had before. So you can obliterate, and this autoreactome does come from B cells. Okay, so everybody's unique, and everybody's a snowflake until they're not. And when they're not, that is the hallmark of disease. So let's take a paraneoplastic example. This is a cancer-driven autoimmune disease called anti-Hu, and the patients are on the y-axis. This is the protein that's the target of anti-Hu encephalitis on the x-axis. And what I would like to point out here is that many of these patients, the majority of these patients with anti-Hu encephalitis, are making the same autoantibody to the same part of the protein. So instead of being all different, they're now all the same. And that is really striking. So it's not random parts of the protein, and there's a lot of reasons why that might be. And so when there's uniqueness, it's the state of health. When there's convergence, it is the state of disease. And we can use that in many respects. So to just sort of hammer that in one more time, here's a disease called APS1. It's a defect in your ability to negatively educate T cells. It's mutations in the AIR transcription factor. Mark Anderson at UCSF worked really hard on this disease. And these patients have a whole litany of autoimmune diseases, everything from Sjogren's to vitiligo to alopecia, growth hormone deficiencies, you name it, they've got it. But it's very heterogeneous. Every person has a different constellation of autoimmune diseases. And we can show in the people with the same clinical phenotype, they have the same autoantibodies that converge on the same targets. So if the patients that have primary ovarian insufficiency are going to have KHDC3L, the RFX6 will be associated with gut dysfunction and so on. So uniqueness is the rule, convergence is the disease, and we can use the convergence to pinpoint a state of disease and underlying targets. Okay, so now we get to COVID. This is the fun part. So this is the work of Aaron Bodansky, a PICU medical fellow in my lab, and this is the story of multisystem inflammatory syndrome in children. And why this matters is because pandemics inherently surface new syndromes that we didn't see before. And one of the things that we often don't talk about is what happens after the pandemic. What about long COVID? And what about this disease? And there are many autoimmune diseases or inflammatory diseases that follow the infection. Think about multiple sclerosis or type 1 diabetes and many others that have epidemiological links to first being infected. So MIS-C came about because of this pandemic. It's a rare disease, 10,000 cases in the US, devastating disease, but when you have millions of infections, you can now see it. So this was a UCSF case, a 15-year-old kid who just had a mild SARS-CoV infection, which is how all these usually go. They resolve the infection, no big deal, but about a month later, they come back to the ICU, or they actually come to the ICU, they're never in there in the first place, and they have incredibly inflammatory disease. Here are some lab values of typical inflammatory markers that are off the charts. This kid and many who get MIS-C require the highest level of supportive care to keep them alive. In this case, the child required ECMO support. That's a clip art of a kid on ECMO. It wasn't the kid at UCSF, but the kid at UCSF was on ECMO and recovered and was ultimately released. So what is MIS-C? It's defined as something that happens to children 3 to 6 weeks after COVID, a total life-threatening multi-organ failure of unknown etiology. The definition is shown below for the CDC case definition. Have to be linked to SARS-CoV-2, have to have fever, evidence of inflammation, involvement in multiple organs. So to be able to study it, you need the patients. So in this case, the cohort was the following. This was by virtue of the fact that we had a great collaboration with Adrian Randolph and the CDC. There's a multi-center collaboration to collect MIS-C cases all across the nation. All these cases were pre-vaccine. I'll say something about that in just a minute. And the outpatient work controls are kids of the same age who got COVID, didn't get MIS-C. And also kids who got severe COVID, but didn't get MIS-C. So 2 different kinds of controls, and then 199 kids who did. And of course, the negative controls. Now, a word about the vaccine. MIS-C, you probably don't hear much about it now because it's almost all gone. It's like dropped off the face of the earth, almost entirely. There are still cases, and according to a CDC report lately, almost all of those cases are kids who didn't get vaccinated. So we do think that the disappearance of MIS-C is tightly linked to vaccination. If you see MIS-C today, guarantee it's almost someone who's never been vaccinated. And it's got to be a kid. Okay, so this is the setup. Again, why do we think that it might be an autoimmune disease? One, it's got unusual activation of T cells. There's HLA-associated component. There's obvious immune dysregulation. There's hundreds of papers published on MIS-C pointing to immune dysregulation, but none with the mechanism. And yet we know that infections and cancer and other things can provoke autoimmune diseases. So given the cohort, we're going to do the technology I just talked about to try to take a stab at this. All right, so 199 cases, the 45 match age controls, the convalescent COVID. We're going to take their serum or plasma, apply them to the full proteome, pull those down, sequence the results. Now, when we ever do this in the lab, the first question I ask my graduate students or postdocs or medical fellows is, look, do the profiles discriminate case from controls? Like we just randomize them in there and blind ourselves, but then when we unblind, can we tell them apart? Because if you can't, well, there's no signal there. And the way we usually do that is don't like sort through Excel spreadsheets and try to use the force or something. You know, we like to use a statistical method, usually machine learning techniques, something like a linear model like logistic regression. And to say, if you can show me that a logistic regression can separate cases from controls with good accuracy and sensitivity, I'll believe there's a signal there. And in the case of MIS-C, absolutely 100%, you can separate cases from controls. They look really different. And it was really driven by 3 different things. This ERFL protein, which, by the way, the repressor of the protein Catherine Blish was just talking about. We should have a little chat later about that. Sorting Nexin 8, another protein highly prevalent in immune cells and chemokine expressing cells, and then an ER-related protein. And so just those three proteins are driving most of the signals here. So this is autoreactivity to peptides in these three proteins. If we just take those three proteins and we make the whole protein and repeat the assay with the whole protein, now not peptides, but the entire thing, we can repeat the result and separate out cases from controls very easily with those three. And so now we're pretty interested in these three, or at least the top two in particular. And in particular, SNCS8, because of its role in chemokine and immune cells, is so clear. And it has a role in antiviral defense, too. So the next question we ask is, well, where is it targeting this? Remember I showed you that data in anti-Hu encephalitis? Is this just some general immune reactivity to SNCS8, or is this something more specific going on here? And it is very specific. So here's a sort of a tiled map of the protein. It's only 465 amino acids. And all the antibodies these kids are making who have MISC, but none in the controls, are to just one little part of the protein. Now, our tiles are 50 amino acids. These are antibodies, so it's something within that 50. It's not the whole 50. So, again, not present in controls, but the patient's appearing to be making the same autoantibody. It's not the same thing. We know that, but it's the same location on the protein. So what do we do to fine map this? We can just turn this technology around and actually just move a little scanning mutagenesis window of alanines, classic old technique, across the target peptide and see where we lose signal. And sure enough, you lose signal right in the middle there. And not to beat a dead horse, we can move stop codons and do single amino acids, and it narrows it down to this one little spot that's shown there in the shading. And that's this MQM-PQG. All right, that's the epitope on this. But how does this relate to SARS-CoV-2? Where does that come into play? So we can play the same game and do this programmable phage across the SARS proteome with MISC versus not, and ask, are they differentially enriched in some way? So the red bars on the top are the reactive peptides in SARS-CoV-2 that are enriched in MISC, in the blue in the control convalescent children. As you could probably see with your eyes right there, there's one region in nucleoprotein that you always see in MISC kids and you never see it in the controls. So what's up with that? Okay, remember this is the SNCS8 peptide. We just repeat that mapping on the NP, the nucleoprotein side of SARS-CoV-2, and it's the same damn thing. So basically, this is convergence. This is what I would call a cross-reactivity between the nucleoprotein of SARS-CoV-2 and SNCS8. And so, okay, but wait a second. You know, SNCS8 is an intracellular antigen. It's not like antibodies going in there and binding SNCS8 inside the cell. So how does this make any sense? And we know there's this HLA association with MISC. There's unusual T-cell expansions. So does this make sense? Well, it could make sense if the antibodies really aren't the thing doing any of the damage. It's the T-cells, and these antibodies are just telling us where to look. So to get at the T-cells, we enlisted our good friend Joe Sabatino at UCSF, and we started with traditional activation-induced marker assays. So we take PBMCs, so take immune cells from MISC patients, we throw in a pool of SNCS8 peptides, so chop up the protein into peptides, throw it in, let those cells, like antigen-presenting cells, B cells, other kinds of cells, take up those peptides or present them on their surface as MHC1 class presentation, and then let the T cells the T cells that are in there, if they recognize something in there, like something presented on MHC class 1, they'll activate and show markers. And those markers can be read out on flow cytometry. That's the AIM assay, essentially. So this is just to get a sense of, do SNCS8 peptides activate T cells? And they do. So, and they activate T cells in MISC, but almost not at all in control. And these are both activating CD4 for CD4 markers and CD8 positive markers. But this is a pool of SNCS8. What about the actual SNCS8 N protein peptides we identified using the phage display? Is it not just the whole protein, but is it one specific part? So now we're going to focus on the cytotoxic CD8 T cells. These are the ones that really do the nasty business. And we have previously established, and I'm not going to show it to you here, that in particular one HLA type, O206, a rather common one, is the most stable with these peptides. And we can load MHC class 1 complexes with both the SNCS8 peptide and the N peptide that we claim are cross-reactive and expose them to cells, sort out the CD8 positive T cells, and then ask, are they both seeing the N protein and the SNCS8? Are the T cells actually cross-reactive? And the data would suggest, yes, they are, but not in controls. That they can see both peptides and they are activated by both peptides in kids with MISC, but not controls. And that's where, like we would have, this would normally we would wash our hands of this, publish it, and walk out the door. But then there's reviewer number 3. And reviewer number 3 says, well, that's all fine, it's flow cytometry and stuff. You haven't actually shown me the real T cell. I want you to go clone the T cell that's cross-reactive, recombinantly express it, and then show it to me again, now one at a time. Okay, fine. I hope that wasn't you, Mark. Okay, so how do we do that? So we get confirmed MISC cases, PBMCs with the known HLA type. We can take their PBMCs and we can expand them or take them right out of the vial, sort the CD8 positive T cells, sort for the ones that are cross-reactive using those tetramers that are loaded with peptide, for the ones that react with both NP and SNCS8, our claimed cross-reactivity, sequence the T cell receptors, find the clones, figure out which ones are unique, and then choose some for stable expression and characterization. And so the results are basically here. There were 259 TCRs that were sequenced in total. And this was with the help of Rob Medelman at St. Jude, a huge help on this project. And we get a network like this. The network means how similar they are in sequence space. So all that, the big circle at the top means all those are basically identical. And we have some from the in vivo expansion as well as just taking it ex vivo, and we have 4 different patients represented here. And so 8 clones were chosen from these different clusters to be expressed individually and then characterized. And I'm going to get to the sort of the magic here. 3 of these clones from 2 different patients, both from 2 different experiments, turned out to be cross-reactive, that they do react to both. The big clone at the top just reacts to SARS-CoV-2 and not self. As you might expect, that's like the T cells that expanded in there. And the individual flow cytometry data is shown here, and a summary of the data at the bottom is shown below. And so what we believe is going on here is shown in the model here, that SARS-CoV-2 infection in kids, MIC is the result of a very rare, but enough to cause 10,000 cases here in the US, of a cross-reactive immune response, wherein they're unlucky enough to acquire a target in NP of COVID that looks like a normally self-expressed peptide, an MHC, that you would normally have tolerance to. And for reasons we don't totally understand, maybe the infection pushes you over the edge, tolerance is broken, autoreactive CD8 positive T cells now see those as the enemy, and you get a widespread inflammation and cytokine storm that is unleashed because of this. That is the model. I put a little tagline there. I mentioned SNCS8. What about ERFL, the repressor of Catherine's protein over there? That guy maps to NP2, and it maps right next to the other one. Interesting. We'll chat about that later. And so with that, I just want to acknowledge this huge group of people that came to solve this. This is a large number of people, not only in my lab, but Paul Thomas's lab at St. Jude, Mark Anderson's labs all around, in hospitals all around the country. The samples came from all the different hospitals and universities shown there in the lower right. And without having a national system to collect these samples and put them all into one place and to bring them under the microscope, we would not be able to solve these diseases. We need coalitions to be able to understand these diseases that emerge as a result of something like a pandemic. And I think that we need to take a sharper look at all the things that happen after infection and how it affects human health in addition to the infection itself. And we can do that with the tools and technologies that we have today, but we have to do a better job organizing ourselves around to do it. So that's the conclusion, and thank you for having me. Peter,
Very, very cool.
you're like an inside ball here, though.
Very, very
it's no
cool.
Fair
Very
answer.
cool.
You answer the question.
So it's a good thing that we didn't put the N protein in the vaccine.
Absolutely. Absolutely. 100%.
It just goes to show that when we're making vaccines, we really want to use what's necessary, but not anything more.
Absolutely. You know, that is, I often don't mention that, but that is something that I think we dodged a bullet on. And protein is one of the most immunodominant proteins in the virus. As we know, when we first profiled it, N protein's off the chart. It's good that we didn't do that.
Do you see any reactivity in people that got the inactivated vaccine? Because the N protein would have been there.
Yeah, we have not been able, we have not had the samples to be able to look at that. But that is a really interesting question. It would have been interesting to look at some of the inactivated vaccine samples compared to mRNA vaccine samples, to look at the profiles relative to this. That would have been fascinating to see.
Yeah, I wondered the same, but
If you've got the samples, I'll analyze them.
there you go.
Okay, game
I
on.
wondered the same, but the CD8 response is presumably going to be very weak or non-existent.
Yeah,
Right.
yeah.
Still. look.
But still, because it's all external protein it's not infecting the cells. Yeah. Yes.
My question was, do you also see this kind of rare autoimmune post-COVID syndrome in adults? And if not, do you have any idea why not?
Well, I think that's an interesting question, and we don't have the answer to it, but just to speculate a little bit here, we know that MIC basically disappeared with vaccination. So being able to push the immune system one way, this sort of antigenic original sin idea, adults have seen a lot of infections, including seasonal coronaviruses, throughout their history. And their immune systems have already been, I showed it to you, have already been molded. They're like locked in. And so I think once you've seen a lot of exposures, the likelihood that you have acquired this target seems to be less for whatever reason. Now there is an MISCA, there is something in adults that does look like this. We have not examined it in the same kind of detail that we have in children. But I do think it's because your immune system's already been influenced in significant ways.
Wonderful presentation. What kind of autoimmune diseases have you seen in the cardiovascular system secondary to this? Thank you.
What kind of autoimmune diseases have we examined in cardiovascular disease? Is that the question?
Yes.
Well, we're super interested in cardiomyopathies and in a variety of different diseases similar to that. And so we're actively seeking, if you've got a good cohort, I'll analyze it. But yes, so it is high on our list. There's some very interesting things to consider there, like in Chagas disease, long-term cardiotoxicity may not be due to the parasite itself, for example. And so I do think not just for COVID, but there's a lot of really interesting work to do in cardiac autoimmune disease and liver autoimmune disease too.
Got it. Thanks.
This may seem a little bit off to the side, but I'm curious whether the phage approach is being applied to chronic fatigue syndrome.
My lab has not. Others may be doing that, but we have not engaged in CFS yet. It's an interesting disease. It's complicated. It's heterogeneous. And we've tended to try to focus on things with really tight clinical definitions and markers. But I don't deny it should be something to be looking at.
There's a lot of classical immunology studies in mice showing that infection, there's even a title, infection breaks tolerance. And looking at it further, and we've done some work on this, it seems like if you just activate the adaptive immune system, antibodies and T cells, you can't break tolerance. But if you also activate the innate immune system at the same time, and you can do that in different ways, then you do. And that also applies to cancer. You know, the people wonder why there are T cells infiltrating tumors that have clonally expanded, but then don't seem to do anything. It's about tolerance, and then you can manipulate that.
Yeah, absolutely. I think the same things are at play for paraneoplastic disease. And, you know, much of what we find in the neurological disease space is paraneoplastic. And they don't even know they had tumors. They beat their tumors. They're long gone.
Yeah, but looking at a whole, like a human population with huge HLA diversity, thousands of alleles, it's a nightmare to try to guess what might happen with any protein. Any protein is going to have some homology to somebody's self-protein, and then might fit into somebody's.
It's okay.
HLA.
I have one slide for you.
Oh, okay. Sure.
That is a great point. How could we have known for just take SNCS8? Would any part of that, we could find a little teeny blast match to the virus? How would we, is there anything we could have done to know that this might be, that part of the SNCS8 might be a problem? Now, of course, not knowing the SARS-CoV-2 sequence, well, that's a problem. But if we knew that, and let me just show you just one slide. I think I stuck it in here. Okay, this is the total processing score. So tap loading immunoproteasome plus HLA MHC class 1 binding of all the 9mers in SNCS8. This is the standard algorithm you can pull off of EIDB, where it combines 3 algorithms into one. All these are negative scores. Nothing is predicted to be on MHC class 1 for HLA-206, except one peptide, and that's it. That is the cross-reactive peptide. So there may be ways we can filter those proteins for what could be predicted to be up there. And I don't think it's an accident. And so I think we have to have a more holistic view of how the immune system shows stuff to the rest of the T cells that are out there and so on to understand this. And it may also explain why there's B-cell, T-cell convergence in this case.
And also, I think it also speaks to, and the caveat is that no vaccine that's given to millions of people will be beneficial for every single person. There's always going to be some collateral damage on some unfortunate person. So that's where the anti-vaxxers come in. They say, oh, look, this kid died, or this kid had a horrible response. Well, that was maybe one kid out of a million.
Yeah.
And so then you have to actually openly discuss the numbers game to say, well, thousands of people benefited, but yeah, one person had a very bad response. So that's, I don't think that's really discussed or known.
No, it's not discussed or known.
You know, it's kind of like hear no evil, see no evil, vaccines are great.
But also on the other side, it leads to misconceptions too. So we've been approached by national blood banks to be able to look at their blood through this technique and other techniques because they are getting requests for blood units from people who are not vaccinated because people are worried about weird autoantibodies from vaccinated people. Well, that's a problem. So you can see where the next level of this goes to. Yeah.
Excellent presentation. Did any of your children have immunosuppression? Immunosuppressed kids more likely. So if you have immunosuppression, do you develop the syndrome or are you prevented from it?
Yeah, so there were no immunosuppressed kids in this cohort. It was filtered for kids with genetic diseases or other immunosuppressions. So these were kids who had normal kids, had normal COVID, recovered normally, but then got very sick.
Right, I'm just wondering whether immunosuppression may protect you from the syndrome.
Well, probably not. So at least for the older individuals that have the autoantibodies to type 1 interferons, they're at massively greater risk for devastating disease or death.
Thank you.
Okay, still on time, right? Yeah. Yeah, great.